The dramatic effect of the COVID-19 pandemic on our planet's global health and prosperity has warranted over 80 pharmaceutical companies from across the globe to develop a vaccine, and 5 have already started human trials. But according to the New York Times, a typical vaccine should not have been cleared for human trials until 2024. So how have these vaccine developers been able to progress so quickly?
The most important factor in the speedy development of a coronavirus vaccine is the use of DNA vaccines, which have never before been commercialized for human use. While traditional vaccines directly expose the human body to parts of a virus that initiate an immune response, DNA vaccines inject a patient with the genetic code responsible for producing these parts. Both methods initiate the production of antibodies that will be available in the future to more effectively and efficiently kill the virus if a person is exposed, but a DNA vaccine can be developed much quicker. DNA vaccines come with many other benefits as well. According to the CDC, compared to traditional vaccines, DNA vaccines can be manufactured, stored, and transported more easily due to DNA's ability to resist temperature extremes, making distribution more practical and less expensive. Because they are easy to manipulate and use generic technology, they provide unprecedented experimentation potential. Most importantly, these vaccines provide a more effective defense as they are able to activate parts of the immune system traditional vaccines cannot.
One company, Inovio Pharmaceuticals, has had tremendous success with the vaccine they developed for Middle East Respiratory Syndrome, otherwise known as MERS. Clinical trials showed that the vaccine provides 100% protection against MERS, further supporting the effectiveness of DNA vaccines. There are many different types of DNA vaccines, and one is the DNA Plasmid, of which Inovio Pharmaceuticals is the leading developer. One method for developing this type of vaccine can be divided into 3 steps:
DNA extraction, plasmid modification/production, and vaccine production. To begin, restriction enzymes, which act as molecular scissors, isolate the DNA that codes for a virus's antigens, and a process called polymerase chain reaction is used to create billions of copies of the sample. Then a plasmid, which is a circular piece of genetic material, is taken from an E. coli bacterium and cut using restriction enzymes. The DNA that codes for the virus's antigens is then inserted with ligase, which can be thought of as molecular glue, and through a process known as transformation, the plasmid can be reinserted into E. coli, which are allowed time to grow as a means to multiply the plasmid. Once enough bacterial colonies form, the plasmids can be extracted and suspended in solution, which will be injected into the skin or muscle of the patient. Soon the body will begin producing antigens, allowing for an immune response to be initiated, leaving behind antibodies that will terminate the virus if the body is ever exposed.
Inovio Pharmaceuticals is likely developing their vaccine with a similar approach and is well underway with human trials. The company is predicting availability by early 2021, and their vaccine is likely to be chosen to develop herd immunity and allow us to return to normal daily life.